Today we're going to take a little bit of notes on osmosis, make sure that we understand osmosis. But first I wanted to do a quick review problem to make sure everybody's on the same page. So it says label the following cells as hyper, hypo, or iso. Draw arrows to show which direction the solute will move in diffusion. Why don't you go ahead and pause the video to think about this. Okay, now that we're back, let's look at this first example. It says there's 90% solute on the outside and 40% solute on the inside. Remember that solute are the particles like salt from our gummy bear example, and that solvent is going to be water. So if I look at this outer thing, and I can draw them with dots, there's going to be a lot more dots on the outside than on the inside. So way more solutes, 90% outside than inside. So I would call this hypertonic. Looking at this one here, it's 50% solute on the outside and 50% inside. If I were to represent that with dots, they would be evenly spaced throughout the entire thing, but I represented it by percentages instead. So I would say that this one is iso. And then again, looking at this last one, 70% inside, if I were to represent this with dots, there'd be a lot more dots on the inside than there would be on the outside because we have 20% solute on the outside and 70% inside. That means that the solution that this cell is sitting in is underconcentrated, so this is hypotonic. So then it says draw arrows to show which direction the solutes would move. So here in diffusion, they would move this way. Here there's they're going to move both ways because there's no net movement, but movement still occurs both in and out. And in this way, they would move out. So you already did an experiment with gummy bears, and today we're going to analyze that. But I wanted to make sure that you had the background knowledge about osmosis first. So osmosis is the passage of water from a region of high water concentration to a region of low water concentration. So instead of thinking about where the dots or the solutes are moving, we need to think about as if the solutes can't move. They can't fit through the membrane. They can't pass through. So what needs to move instead? Water. You might have experienced this if you've ever made like a saltwater solution before to gargle or something like that. If you put too much salt in the salt water, it's not like you can take the salt out. The only way that you can dilute it and make those molecules more spread out, have it taste less salty, is to add more water. The idea behind osmosis is that same thing. So let's look at these two osmotic tubes. That's what these little tubes are called here. And you can see that there's this selectively permeable membrane between. Now the solutes, as you can see, are too big to fit through the membrane. But here we have less solute and here we have more solute. The solutes still want to be as spread out as they can be, but since they can't move, the water has to move. So here we have a higher water and here we have less water. So the water is going to leave this area and move into this side. What that does is it actually lowers the water from here and raises it on this side until you can see that the solutes are evenly spread out on both sides. So you can see that there's even distribution of solutes because the water moved from where there was more water to where there is less water. So now we're going to take a look at what happens to plant and animal cells in a hypertonic, hypotonic, and isotonic environment. So we have to first think about what's that main difference between plant and animal cells? And the main one that is going to be important for osmosis is this cell wall. Plants have cell walls. Yes, plants have chloroplasts and animal cells don't, but in terms of osmosis, the thing that's going to be the most important difference is that plant cells have cell walls. So let's take a look at this example. Again, looking at this top part here with the water, here there's lots of water, right? High concentration of water. And here there's a lower concentration of water. So if you'll notice, the water is going from where there's lots of water to where there's less water. And that's because the solutes can't move instead. Notice it's the opposite direction of the way that the solutes would move, but since the solutes are stuck and the water is moving instead, the water needs to go from area of high concentration of water to an area of low concentration of water. So what does that do to the cell? Well, here is a drawing of a red blood cell in an isotonic solution. So that's a red blood cell looking pretty nice. You have water going in, you have water going out, you have isotonic conditions because the solutes inside are the same as the solutes outside. Now look at what happens when you have a hypertonic solution. Hypertonic means, again, that there's more solutes outside than in, so there's more water inside the cell. So the water is going to want to go out through osmosis. So you have more water going out than you do going in. When you have more water going out, you end up with this kind of shriveled cell. So that's what happens in a hypertonic solution. Hypotonic would be that there's way more water outside than inside, right? So in this case, you're going to have water going in. And what this is trying to show you is eventually the cells will get plump and plump and plump and plump and then explode. So here's the bursting cell. Let's take a look at a different drawing and then what they look like under the microscope. So here we have hyperosmotic. So again, it's in a hypertonic solution. Here's isotonic. Here's hypoosmotic or hypotonic solution. So this is red blood cells sitting in concentrated salt water, and you can see these cells here are all very, very shriveled. This is what red blood cells look normally, and this one here, red blood cells have this kind of like lovely donut shape like you see in this one here, but taking a look at this one, it's getting super fat. So it's getting overly full here. So this is red blood cells starting to get very, very full. Not quite bursting yet, but which is what's happening in this picture. But eventually, if they sit in distilled water, they will continue to explode. Another drawing, just in case that helped. What I like about this one is it shows the way that the water flows out when it's hypertonic, back and forth when it's isotonic, and in when it's hypotonic. So now predict what will happen in plant cells when placed in hyper, hypo, and isotonic. You know about what happens in animal cells. We looked at human red blood cells, but what about plant cells with their rigid cell wall? Why don't you pause the video a minute to think about it? So this is what we have going on here. So this is your isotonic cell up here, and you can see that it's got all this great pressure. This is this large vacuole. This right here is the large vacuole. Then you can see here in a hypotonic solution that yes, it fills with a bunch of water and that vacuole is very full and it's causing a lot of pressure on the cell, but the cell stiffens and generally retains its shape. However, if you place it in a concentrated salt solution, look at what happens here. The cell membrane actually shrinks away from the cell wall, which is no good. When this happens, the cell dies. So in this hypertonic solution, we have the cell membrane pulling away from the cell wall. So here we have hypotonic and hypertonic in the plant cell, and you can see the cell wall here, and it's keeping its shape. You've got a lot of water pressure from having all of the vacuole full, but the cell wall can handle it, so it's okay. And you can kind of tell where everything is by looking at the chloroplasts. Now here, if you look at the hypertonic, you can see this area right here, that's the cell membrane because you can see all the chloroplasts in it. So it's totally what's called plasmolyzed. It's moved away from the cell wall, and this would result in cell death. So this is showing a picture of what the cell membrane would look like right here, like it's barely holding on to the edge of the cell wall. Couple of more scope pictures. Isotonic solution. This is what a plant, this is a water plant looks like, looking at one layer of cells under a microscope, so isotonic. Hypotonic, it looks pretty much the same. It's just the cells are a little bit more full, so they're harder to focus. So you get a little bit of that blur. And then you'll see in the hypertonic, you see the cell wall is intact, but the membrane has pulled away from it. Okay, self-quiz time for a little review. Describe what will happen to an animal cell in a hypertonic, isotonic, and hypotonic solution. Which is ideal for an animal cell? Then describe what will happen to a plant cell in each and which is ideal. Why don't you pause the video to see if you can answer this? So here's the answer. We have an animal cell on the top frames. So you have an animal cell that's all shriveled up. In isotonic, they look normal. In hypotonic, they're lysing open, splitting open. So normal is our best bet here for the animal cell. The plant cell, however, hypertonic, we know that's not great because it's plasmolyzed, cell death. The cell membrane has pulled away from the wall. But isotonic, it uses this word to describe it, flaccid. The best way I can think about this is celery that's been in the fridge for like a week and a half, like it's super limp and it feels really bendy. That's flaccid. As in it doesn't have as much water as it should to keep that cell wall firm. Turgid is filled with water. So this is when you have like a nice snap when you bite into celery. That would be an example of turgid. So plant cells actually prefer to be filled with water in a hypotonic solution because the water helps give the plant support as well. That's it for our osmosis notes. Now we're going to look at the results of our lab. Okay, so I've headed over to my OneNote, and right now I'm in the Gummy Bear Osmosis Lab questions. You've already done part 1 and part 2, and then I put this little section here, answer the questions below after conducting the experiment. So this is where you should be at number 6. So I want you to go ahead and measure the length, the width, and the depth of the gummy bear, calculate the final volume just like you did previously in class from the initial, and then do the final minus starting volume so that you get a difference in volume. Now, you may have one gummy bear that's so large, it's slippery and slimy and difficult to measure. Do the very best you can with that gummy bear. Again, the best you can at measuring. Then answer the question, did your observations match your prediction? If no, explain what you think accounts for the difference. So part 3 is looking at osmosis in animal cells. So there's a lot to read here. And then you're talking about blood cells and different salt solutions. So your job is to read the material and then by looking at these drawings, figure out what percentage salt solution are these red blood cells sitting in? And you will have to read to figure that out. And then here where it says tonicity, you're going to write whether it's hyper, hypo, or iso. Then answer these 2 questions. In part 4, you're going to draw pictures of plant cells, one that's turgid, one that's flaccid, and one that's plasmolyzed. That sounds very familiar, like the drawings I perhaps just showed you. Then you want to indicate with arrows the direction of the water movement across the membrane, so be sure to use the arrows. And then here again, tonicity, label whether it was hyper, hypo, or iso. And that's it for the lab.